Rudolf Virchow, a German physician, said, "Where a cell exists, there must have been a pre-existing cell, just as the animal arises only from an animal and the plant only from a plant." He summarized this concept by saying, "Every cell from a cell. Cell division is essential for the continuity of life. Cell division is when a cell replicates itself. This is important for growth, development, repair, and reproduction of eukaryotic organisms." In order for a parent cell to divide to create two daughter cells, it must undergo the cell cycle, which is a series of steps that ultimately leads to cell division. The cycle consists of the interphase and the M phase. The interphase accounts for 90% of the cycle. During this time, a cell prepares for division by growing and copying its chromosomes. Interphase is made up of three subphases, including the first gap, G1, S phase, synthesis, and the second gap, G2. During these subphases, a cell is growing and duplicating organelles in preparation for division. To summarize a cell cycle, a cell grows during G1, duplicates its DNA, and continues to grow during the S phase, then grows more as it prepares for division, G2, and finally divides in the M phase. The mitotic phase, which accounts for the other 10% of the cell cycle, contains mitosis and cytokinesis. Furthermore, mitosis is broken down into 5 stages: prophase, prometaphase, metaphase, anaphase, and telophase. Cytokinesis then finishes. During prophase, a mitotic spindle made of microtubules begins forming from the centrosomes. This is where they are at opposite poles. Then the asters extend, the nucleolus disappears, and chromatin condenses into chromosomes. During prometaphase, a nuclear envelope dismantles and chromosomes form 2 kinetochores. These are structures of proteins that attach to the centromere and act as a port for the microtubule fibers. Microtubules begin attaching to the chromosomes, becoming kinetochore microtubules, which move the chromosomes. During metaphase, the chromosomes align on the metaphase plate, a spot that is equidistant from the two sides. And during anaphase, the chromosomes split apart, and the chromatids are pulled opposite directions by the microtubule fibers. And while this is happening, the cell begins to elongate. By the end of anaphase, the cell is to complete collections of chromosomes at either end. During telophase, a new nuclear envelope is formed. Chromosomes unfold back into chromatin, nuclei reform, and the cell continues to elongate. And during cytokinesis, the two daughter cells divide at the cleavage furrow. There are a few differences between that in the M phase when comparing plant and animal cells. For example, plant cells don't have centrioles. They can still perform mitosis without them. Also, during cytokinesis, rather than forming a cleavage furrow, the plant cells form a cell plate, which becomes a cell wall dividing the two daughter cells. To regulate cell division during mitosis, there is a cell cycle control system, and in this cycle, there are checkpoints during the different phases of the cell cycle. A checkpoint is a control point where stop and go-ahead signals regulate the cell cycle. There are 3 big checkpoints: G1, G2, and M checkpoints. The G1 checkpoint, known as the restriction point, is considered the most important. If a cell passes a G1 checkpoint, it continues down the cycle, and if it does not, it goes into the G0 phase, which is a non-dividing state. A cell knows when it should divide when the cyclins, a type of protein, accumulate to a critical number. CDK, cyclin-dependent kinase, is a chemical that speeds up cellular reactions. As said in the name, it is dependent on cyclin. As time goes on, cyclin accumulates. After hitting a critical number, the cyclin binds with CDKs, creating a complex like MPF, acting upon the cell and pushing it through mitosis. MPF then disappears, allowing the cycle to restart. Internal and external signaling affect when a cell divides. For example, at the M phase checkpoint, chromosomes separate. Does not occur unless the spindles are properly attached to chromosomes. When it is attached, it is able to separate. Another thing affecting are growth factors, a protein released by cell telling other cells to divide. There is also density-dependent inhibition, where cells touching in a crowded space tell each other to stop dividing. There are cells that do not follow the rule, like ones that divide extensively. These can be caused by a number of things like mutation in genes. Cancer cells continue to divide in the absence of growth factors. There are also cases of abnormal cell control system, where the checkpoint don't function as they are intended to. Transformation is when a normal cell becomes a cancer cell. If a